Okay, guys, back to finish off the protein synthesis discussion. Mutations are what we're going to get into today. So hope your day is going well. And let's get in here. So if you recall, we now have some knowledge on transcription, conversion of DNA into the mRNA molecule. That is accomplished by that little very dynamic enzyme that we call RNA polymerase. So all that happening in the nucleus, right, transcription. Then we move out, the messenger RNA travels out, and with the assistance of the ribosome, with the assistance of the tRNA molecules, we will complete translation, which means we're going to build our primary protein structure. So you have that little worksheet, where you get practice with vocabulary. I gave you a strand of DNA and asked you to transcribe and translate. And I hope you were able to navigate through that. Well, now we're going to look at an important evolutionary aspect called mutations. So mutations are any change in the DNA molecule. So any change. It can be a huge traumatic damage to the DNA, or it can be a very minimal, simple change to that DNA molecule. But any change qualifies as a mutation. So mutations are considered rare events. They're not just going to happen spontaneously most of the time. They are triggered by certain things such as radiation, certain mutagenic chemicals, viruses, right? The virus itself takes its DNA and crams it into your DNA sequence, so causing the complications there. So any and all viral infections are technically mutations, and it changes the DNA. And if we change the DNA, we may change the RNA. And if we change the RNA, we may change the protein structure, the primary structure. If we change the primary structure, we're going to change the secondary and the tertiary structure. And if we change the tertiary structure, we change the way that protein functions. So it's like a domino effect, all triggered by that change in the DNA. So the most common type of mutation is something we call a point mutation, right? Point mutation. Point mutations are going to involve one nucleotide, one adenine or one thymine or one guanine or one cytosine, one nucleotide. Once there's a mutation, it's usually considered to be detrimental, something bad, but sometimes the mutation will be neutral. It's neither good nor bad. And sometimes in rare situations, that mutation might trigger something good, it may change the way a protein functions and makes it a little bit more optimal. Right? So this is a good setting to talk about this. So we know now that, for example, as far as the virus, the COVID virus is concerned, so it had its genetic sequence to affect possibly bats, right? Now, when that mutation happened, it made the virus be able to jump into human hosts, right? Well, that's very detrimental to us, but as far as the virus is concerned, wow, that's good. I can now go and infect all this new, these countless people on planet Earth, right? So this idea of the mutation depends on the perspective, may be bad, may have no effect, or it may be good. Depends on the situation, depends on the DNA that's being mutated, and depends on the sort of the environment in which that mutation takes place as well. So we're going to look at 4 types of mutations: silent, missense, nonsense, and frame shift. Now, if I can have you on these notes, sort of circle, sort of highlight, sort of make a distinction that these 3, silent, missense, and nonsense, silent, missense, and nonsense are point mutations, right? So point mutations. Frame shift mutation is something different. A frame shift mutation is not a point mutation. So what does it mean to be a point mutation? It means that we're only affecting one nucleotide, right? And point mutations are considered to be substitution mutations. So let's say in sports, somebody gets injured. Well, that injured person comes out and somebody goes and replaces them. So the overall number of players on the team, players on the court, on the field, remains the same. So the number of players doesn't change. In a point mutation, we take out a base, but another one is put into its for its replacement, into its spot there, right? So point mutation, also known as substitution mutations. Silent, missense, and nonsense mutations are all categories of point mutations. A frame shift is not a point mutation. That's something else, and I'll get to that a little bit later in this discussion here. So by definition, we know a mutation is any change to the DNA sequence. Well, if we have a change, if our DNA changes, our RNA changes, but there's no corresponding change to the amino acid. That is known as a silent mutation. So for example, we have a certain DNA that gives us this RNA sequence, and we're going to focus on this codon right here, on the messenger RNA, GGC. GGC, we come back over here, GGC. So GGC. GGC is going to code for glycine, right? GGC. GGC coding for glycine. So that's what we're supposed to have in that particular sequence of that amino acid chain. Well, we have a silent mutation. So we have a change in the DNA first, which affects the RNA, right? And now instead of the correct GGC, we ended up with a GGU. Oh, we're not supposed to have uracil there, right? So that means the original G in the DNA was somehow mistakenly damaged. Oh, there's nothing close, you're the closest thing nearby, and the wrong A gets crammed into that location. So instead of CCG in the DNA, we have now CCA in the DNA. So the U instead of the C. So we have now GGU. Oh, so we have a change in the mRNA, GGU. Ooh, but luckily GGU codes for glycine, right? So whew, we dodged the bullet there, yeah? So there was a mutation, a change in the DNA, a corresponding change in the RNA, but no corresponding change in the amino acid chain, right? So we have glycine where we're supposed to. We had the mutation, we still have glycine where we're supposed to. So why do we call it silent? Because we won't even know that the mutation happened, right? There's no symptoms, there's no effects. The protein will be built like it's supposed to, and that protein will function like it's supposed to. So one, is not necessarily a good situation that the mutations are happening, but if you have to have a mutation, you hope it's a silent mutation, right? Because there's no biological effect. And going back over here to our codon chart, I hope you understand sort of our DNA's kind of insurance plan, little backup plan. As long as the first two letters stay the same, usually, not all the time, but usually if the first two letters of the codon are the same, we get the same amino acid. Right? CCU, CUU, CUC, CUA, CUG, all coding for leucine. So the third letter in this situation doesn't matter. Here, the third letter doesn't matter. Here, the third letter didn't matter. Sometimes that's not the case, right? So here we have AGA. Well, if we change the third letter, AGC, ooh, it does change the amino acid, right? So it's just, it works sometimes. It's a little insurance plan. Sometimes it doesn't hold true, right? But the moral of the story, take care of your DNA. You don't want it to be mutating, right? You don't want to be exposed to all this radiation, all these mutagenic chemicals. And it's just a matter of chance, a matter of luck, which of these bases gets attacked. When the virus crams in there, the virus isn't picky. The virus is just going to squeak in wherever it can. So if there's a mutation, we hope it's on that third letter of the codon, right? We have no say in the matter. It's just chance. I hope that made sense with our silent mutation. Now, a missense mutation, again, if you notice, we're not changing the third letter in the codon, we're altering the second letter. So in this situation, if we should have CTT, the normal healthy transcriptions should be GAA. GAA, if you look at the codon chart, will give us glutamine. Well, we have a damage, a point mutation, a substitution mutation. We have the wrong nucleotide that's trying to repair the damage, CAT, which gives us GUA, which codes for the incorrect valine, right? So, oh man, now this is no longer silent. There is going to be a significant impact on our primary protein structure and the overall functionality of that protein. So, I don't know if you've heard of the genetic condition sickle cell anemia, right? It's a blood disorder that makes the blood cells very fragile. Those proteins are not very sound and they break very easily. Well, why do they do that? Well, because instead of the normal amino acid, we have the wrong one. And it makes that point very fragile and very weak on that protein. So there is a biological effect, and in this situation, it's not a good biological effect. So missense causes an amino acid substitution, right? First, it caused the point mutation, a nucleotide substitution, and that caused the wrong mRNA, and then ultimately the wrong amino acid. So the wrong amino acid is set forth, and that caused the change in the protein shape and the change in the protein function. There's a lot of genetic diseases that work this way. So again, missense, so biologically significant, yeah? Now, are all missense mutations good? Or are all missense mutations bad? Or are all missense mutations neutral? It depends. It depends on the situation. In this situation, this frog, I don't know all the story, but frogs aren't supposed to have this many legs, right? So we know there was a mutation that had an impact early in embryonic development that caused multiple legs to form. So well, more legs, which should make it hop better or swim faster, but it doesn't seem to be the case. These legs are not really functional, probably slowed down the frog, right? So in that situation, caused an early death in the frog. We're going to say that missense mutation was a negative, a detrimental type of situation. Over here, we say, well, normally cheetahs are supposed to be spotted, but this cheetah has some sort of mutation in the fur coloration that makes the cheetah striped instead of spotted. So is that a bad thing? Is that a no effect thing? Is it a good thing? Right? I don't know. I don't think it makes it any faster. If it makes it harder to camouflage, then we'll say, well, yeah, maybe that's going to be harder for this cheetah to sneak up on prey, right? Maybe it helps it camouflage better, then that would be a beneficial type of situation, right? So it depends on the environmental sort of context in which the mutation takes place. And now if you look at this child, right? Oh, look at this little mutated child here, yeah? So mutated child because first, very fair skin, and second, blue eyes, right? Both of those are due to missense mutations. The quote-unquote wild type of normal human phenotype, of how humans appear physically. The wild type is considered dark eyes, dark brown eyes, and a darker skin color. So any deviation from that is considered to be a missense derived sort of physicality, right? And again, blue eyes, right? I don't have blue eyes. I don't know if blue eyes make it harder to, drive into when the sun is coming into your eyes, right? Does blue eyes make light refraction more? I don't know, but if it does, then we say, well, that's not very good. Does light skin sunburn easier? Well, maybe that might not be a good thing. Could blue eyes make a baby cuter? Could blue eyes make a person more beautiful, more handsome, enhance the chance that they're gonna, get asked on a date or find a partner, that kind of stuff. So if there's some benefit conveyed by that mutation, we say that that missense mutation was beneficial. If there's no effect, well, no effect. If it makes it easier for them to get sunburn, if they don't have if their eyesight is sort of diminished in any way because of the blue eyes, then we say, well, that's not a good thing. But again, it's all contextual, depends on the situation the organism finds itself. Right? So the missense mutations. Okay, a nonsense mutation. A nonsense mutation, this is where, again, I hope this makes sense. A nonsense mutation is a missense mutation. Again, a nonsense mutation is a type of missense mutation, but it's a very specific type, right? So we're going to change the DNA, we're going to change the RNA, and we are going to change the amino acid. But in this situation, we end up with one of the three stop instructions, right? So all we did was change one letter in the DNA, we change one letter in the RNA, and it ends up accidentally making a stop, right? A stop instruction. So we substitute a premature stop codon, which is coding for, it's being translated to one of the three stop commands. So basically we should have had all of these amino acids here, but we got the stop instruction too soon. So we started, we stopped, and that rest of the protein was not completed. So that incomplete protein is released out to do the job, and it's not going to be able to do the job because it's not functional, it's not complete. Nonsense mutations range from not too bad to very, very significant. Right? So I don't know if you've seen people with the condition vitiligo. So here the normal skin pigmentation is starting to be formed, and then we have some mutation throughout life that terminates that production of the skin. Some people are born with the condition. Some people develop the condition through mutation later in life. But as far as mutations go, there is no biological harm caused by the vitiligo condition. There may be some psychological, maybe being very self-conscious side effects of that condition, but again, that is caused by an early stop. So the skin pigment protein stops being produced too soon, and it doesn't then color the skin as it normally should. It happens in humans, happens in animals. So we see these animals, this peacock here with that missing coloration. Again, not so bad, maybe, but in humans, we also see effects of nonsense mutation. We know that the lip should be complete, that embryonic development, during that process of embryonic development, that amino acid is cut short. So instead of completing all of that tissue on the palate, on the lip, those proteins are not formed, and the child is born with that cleft lip, cleft palate. And again, it gets worse. Sometimes you'll see babies born without limbs, babies born without organs, babies born without eyes, because of those mutations. Maybe mom or dad didn't take care of themselves. The sperm and egg could have been mutated, and then the child doesn't develop the way it should. A lot of natural miscarriages happen because of nonsense mutations as well. So take care of your DNA. You don't want these types of effects in your body or in your children's bodies. Yeah. All right, so to summarize, let me back up way over here. So 3 types of mutations, right? So we have the silent, missense, nonsense, which are substitution or point mutations. Silent, no biological effect. Missense, a biological effect. And nonsense, which is a type of missense with an early stop, which typically is very detrimental. So the last but not least is our frame shift. So the frame shift mutation, right? Frame shift is an insertion or a deletion, right? So we're going to insert or delete, but we don't adjust the number. So in sports, an extra player is added in and they get to play the rest of the game there, or a member is removed and we don't replace that player. So it changes the number of individuals on the court, on the field. A frame shift mutation is going to either add a nucleotide or it removes a nucleotide, but we don't get the number of nucleotides back to its normal configuration. So again, this type of mutation shifts the code, changing many of the codons to different codons. And it almost always leads to the production of non-functional proteins. So not good. So again, let's see where are we here. So we're going to have, at the DNA level, there's a change. So we should have had here at the DNA level, AAA. So AAA, which would transcribe to UUU, but somehow there was a deletion. AAA, we lost that third A, right? So now we have AA. So that causes then the loss of this third letter of this codon. So now we have UU, and then we have to shift over to the next codon to borrow that letter. So instead of having UUU, now we go UU and we borrow the G from this codon. So instead of producing phenylalanine, now we produce leucine. And it again is incorrect. The ribosome will shift the letter to the left or to the right, depending if it's an insertion or deletion, and it's going to skew everything incorrectly after that after that frame shift mutation. So again, not good. And sometimes we don't end up with a stop. Sometimes we do end up with a stop. Sometimes the stop is early. Sometimes it just makes a random mess of the situation. So I hope that makes sense. Let me get out of this thing here and see if I can open up that document over here. So again, I hope all this mutation stuff makes sense. It has a profound effect on biology, on the evolutionary process. So now that you have a little bit of knowledge on the vocabulary, we've covered the silent missense nonsense, the point mutations, and then that separate frame shift mutation. So I would like for you to try to fill in the rest of this study guide. Fill it out, send it back to me, and it'll be for bonus points in lecture. I think I mentioned something about lab. I don't want to, kind of mess up any of Mr. Benavides' lab grades or anything like that. So complete this, and it will be then for bonus points for your lecture course, 1306. So you're always going to use the basis of this DNA strand. Keep track of the codons, right? So after you construct your codons, designate the first codon, 1, 2, 3, underline, 1, 2, 3, underline, 1, 2, 3, underline, 1, 2, 3, under, and basically generate your codons and see if you can generate that mutation there, right? See if you can be like a genetic engineer and get in there and alter the DNA sequence first to see what effect it will have on the RNA sequence, and then lastly on the amino acid. So 3 is asking for the silent mutation. 4 is asking for an example of a missense mutation. 5 is asking for an example of a nonsense mutation. And 6 is going to be a little bit messy, but maybe somehow you can maybe copy. I guess I could do this. Maybe copy this strand, paste it down here. Might need more space, but paste it down there, and then show that that frame shift. Either add a letter or remove a letter, and don't substitute. Retranscribe, retranslate, and see how different that amino acid sequence is to the normal one. they kind of ask you some of this stuff maybe on the homework, but again, the location, where do these things happen? We talked about the role of RNA polymerase, the role of the ribosomes, and then we get into some sort of mutation evolutionary concepts here. Yeah, so again, extra credit, you don't have to do it. It won't count against you. But if it helps, gives you more practice, by all means, go for it, yeah?